Thank you so much, everyone. My name is Shashank Ashley. As you might have heard for one of my presentation early on, I'm one of the organizer, and I am glad to have this opportunity to chair this session. And in this session, we have Dr. Pele and Dr. Mercier, and then Dr. Vinod Kumar. Uh, Dr. Pillai is one of the foremost expert in aging research, and he worked for a long time in the longevity groups. And specifically, he is into translational side. I am excited to listen to his talk. His research focus on aging and regeneration, advancing new insights into age-related health interventions. As I mentioned, this is going to be one of the translational side of the conversation. With that, Dr. Pillai, please go ahead.
Thank you very much. And hello, everyone. So first of all, thank you for inviting me to speak at this conference. I'm going to share my screen right now. And hopefully you can see it, I believe. And I will begin presentation mode. Right. So yeah, I'm a scientist. I'm a biologist, expert in the biology of aging. I've been working in this field for over 15 years. And I've also moved toward industry and created my own companies about 5 years ago. And so my interest was, of course, to turn all of my research in the biology of aging into product, because my goal is to try and find ways to either slow down or even reverse aging. And so the topic of today is skin rejuvenation, and how skin rejuvenation can be a model for basically whole body, all organs rejuvenation in the future. So this question that I typed here, can we reverse the aging process, is a question that I'm obsessed with for a very long time, almost 30 years, which is a lot for someone who is only 45. And basically, my interest is not just to try and slow down the process of aging. I mean, in the previous talks, we heard about several age-related disease and how prevention is very important to try and slow it down. And of course, I care very much about that, but I'm also very much into the possibility of actually reversing aging at different scales. And so my view of aging is that basically aging is as complex as biology itself. And so on this little graph here, you can see that, yeah, biology is extremely complex, because it's made of different elements at different scale, all of which are interacting with each other. And that makes an incredibly complex network. And every single of these elements, whether it's molecular, macromolecular, organelle-like, cellular, or even at the level of organs, they all can be considered as units that can be either considered young, fully functional, or that can turn old with time, which means dysfunctional, damage in many ways. And that would be called aging. And the possibility of going backward is what we call rejuvenation, right? So there's two possible strategies against this. One is to try and slow down the aging process by prevention or protection of these specific elements. And the other is to either repair or degrade the old element to build new ones. So if you look at this at different scale, like here, there's example, for example, proteins that are, of course, crucial for biology in general at every aspect of it. They do get oxidized, and that's their own aging, basically. And this could be protected by antioxidants or chaperones. But it can also, since proteins work as a population, you could just degrade them, and then new one will be appearing, and the population of protein will be younger. And the same thing can be seen for mitochondria, where you can either reduce their damage or basically destroy them through mitophagy and get a younger population of mitochondria. And the same thing applies to cell as well. So the question is, can we apply this process, these ideas, on entire organs. And my research, especially in the context of Bhimini biotech, was to start with skin. So there's a reason for that. There's several reasons actually. And it sounds less important than all the diseases that have been presented until now, right? Skin seems like it's more of a beauty problem, but actually, of course, it's not, because skin is the first layer of defense against the environment. It's also connected with many other systems, including the immune system, the endocrine system, and basically it has multiple roles. So aging of the skin has consequences. But I mean, beauty matters as well, especially if you try to motivate people to enter into this, consciousness of being careful of your health. Well, sometimes the mirror is the best way to motivate you. Like, you see that you're aging, that gives you motivation to be more careful about many aspects of your lifestyle and to improve your health. Now, more concretely, skin aging, well, skin is made of three layers, right? The epidermis, dermis, and hypodermis. And epidermis is basically just a wall to protect us from the environment. And the dermis is the part that give the texture of the skin, its elasticity, and actually it plays a major role in many different aspects. And the main cell type in the skin are fibroblasts. And so these cells, they are like little factories within the skin that produce constantly collagen, elastin, and hyaluronic acid, just to mention a few, which basically create what we call the matrix of the cell. And the collagen, for example, is responsible for the texture of the cells, the elastin for the elasticity. Hyaluronic acid is more involved in the hydration level of the skin. And with age, we lose those fibroblasts. And so it seems that, of course, by losing this fibroblast, plus with all the damage that accumulate, the skin is getting worse and worse. And so it gets, wrinkles start to appear. There's loss of elasticity that you can see in all skin types, well, differently, but you can see it anyway. And dehydration, the skin just gets drier and less healthy. And of course, its role as a barrier is also decreasing with all these elements. So the solution that could be solving that, at least in big part, is to re-inject fibroblasts. So there's a technology called autologous fibroblast injection. I did not invent it. It was invented over 25 years ago. And the idea is pretty simple, is that since we lose fibroblasts with age, why don't we, well, simply grow some and inject them back to replace the one that were lost and to resynthesize this matrix to recreate the collagen and hyaluronic acid and elastin and all the other elements that are going away. And the process itself is not that complicated, right? You just start with a small skin biopsy, usually a few millimeter diameter is enough, from which you extract the fibroblasts and you grow them in culture. So you start with only, there's basically in such a small fraction, there might be only a few hundreds of fibroblasts that are able to divide. And from these few hundreds, you reach millions of them, up to 50, 60, 70 millions, that you can then inject back into the skin. And so they will replace the missing one. And so with our company, we developed what we call AFI Plus. So we didn't go very far with the name. It's just an improved version of this autologous fibroblast injection, in which we took every single aspect of it and optimized the process. That's the first thing. We managed to get faster cultivation, better and less painful injection. Well, we really focus on different aspects. But of course, the most important part is to get healthier and younger cells. So the classical approach of autologous fibroblast injection doesn't really care about how healthy the cells are. And not only do we lose cells with age, but of course, those cells also tend to get older and less efficient at what they're doing. So being able to get younger cells will improve dramatically this technique. So we work on that, and we have different, let's say, combination of technique to improve the health and use of these cells. And one of them, and the one I wanted to briefly describe here, is based on the concept of cellular heterogeneity and selection. So what does it mean? It means that within a cell population, even if you just look at one cell type, here fibroblast, so the picture in the back is not fibroblast, but it's just for illustration. These cells are not all the same. So you always have heterogeneity, meaning that some of the cells are younger, some are older. Some are older to the point they are senescent and they start, degrading. So that's another problem. These one are not too problematic for us because senescent cells don't divide. So I won't have that in culture, obviously. But there are clearly still some cells that are much healthier than others. And so the idea of selecting for these cells at the very early stage of the cultivation allow to get a final pool of cells that are actually much better than if you didn't go through this process of selection. So I'm not going into too much details about the other aspect of our cellular rejuvenation process, but in terms of results, when you look at the cells, with this improved cultivation process, what we see is that the cellular fitness, which means the speed at which cells grow in culture, which is known to decrease with the age donor. So if you got cells from someone who is 70 years old, the cell won't divide as fast as someone who is 20. And we managed to improve this cellular fitness by up to 84% in our approach. We also check for stress response, which is very important as well, and also something that is lost with age. So we take cells, we stress them, and we see how well they respond, in that case, by expressing proteasome. And we've seen also a pretty impressive improvement in this. So not back to 100%, but still pretty good. And we also looked at mitochondrial health because mitochondria are considered as one of the key clock of aging. So, it's very important that these mitochondria are also healthy to get better fibroblasts, which will do their job for longer. In terms of skin effect, so on the left here, you got results from the traditional method, the AFI, without modification. And I put it there because, well, we can still see, of course, improvement, but most importantly, we can see that with such approach, you can rejuvenate skin and it will last for a very long time. So that's something that people are interested when they do this kind of treatment. If you do Botox treatment, it's purely cosmetic. It's purely the appearance of the skin that is improved, but the skin hasn't get any younger. It just looks younger. But our goal, of course, is to make the skin biologically younger. And you can see that even in the original method, because even 3 years after the injection, the rejuvenation still stand, and that's very important. So we've done our own, of course, result from our own lab. And we see also like very dramatic, in some cases at least, very dramatic reduction in wrinkle and in skin sagging. Here are examples in the neck and décolleté. This is the same person, and she's 70 years old. And after 3 months only, her skin is back almost like when she was 50. So that's a 20 years rejuvenation. This is pretty good. We have other similar example, but what we also seen so far with just one case, so it's anecdotal, is that we also managed to get very good results with melasma. And that was to some degree unexpected. And it underlined the fact that it's not just about wrinkles. It's about the overall health of the skin. And that's really our goal. So we're getting interesting results. We're trying to also go deeper and see if there's more aspect of aging that can be of skin aging that can be improved. The method itself, we're working on ways to improve it further. But our goal for the future is to also start looking into other cell types and trying to use this rejuvenation approach to other organs. So to do so, our first, let's say, target after skin, obviously, will be mesenchymal stem cells, eventually hemopoietic stem cells, and eventually other cell type that we are interested in. And so being able to, I mean, this stem cell treatment already exists, but we will have the possibility to improve even further the rejuvenation and the positive aspect of all this treatment. So that's pretty much it for me. That's actually much faster than I expected.
Thank you, Dr. Pellet. Thank you so much.